Welcome to MOOC course on Introduction to Proteogenomics. In the course so far, the emphasis was to give you the good understanding of genomics and proteomics, different basic concepts and tools available to do data analysis. But now we will shift gear and move on how to integrate data from both genomics and proteomics in the form of proteogenomics, because neither the genomic or proteomic platforms could provide you the complete picture. Proteogenomic technologies, proteogenomic tools have power to combine various genomic approaches along with proteomic data sets to provide a comprehensive, very broad overview at transcriptional and translational level. In today's lecture, Professor David Fenio will introduce you to the basic concepts of proteogenomics. So, let us welcome Dr. Fenio for his lecture.
So far you have had first an introduction to genomics, then to proteomics and also to machine learning, and now I will try to give an introduction to proteogenomics, where we combine the genomics and the proteomics and see what additional things we can do when we have both kinds of data. So as you heard from Carl, when we often when we want to, the first step in proteomics is to identify peptides and then proteins. So one very important thing with this database search is that if the exact protein sequence that we're looking for is not in the database, we won't be able to find it. So what one can do then, and of course we know that in cancer there are a lot of changes in the genome, and then these could lead to changes in protein sequence. But if we just use the reference protein sequence database, we won't be able to identify these. But now, since we have both genomics and proteomics data, we can use the genomics data to modify our database, to add in all the effects of the genomic changes. So that's, and then we make a more comprehensive database that then we should be able to see the specific changes that happen in this tumor. So, for example, just to look at a few examples, if we have a single nucleotide change, like in this case here, we have one base that's changing that then can lead to a change in an amino acid. So this is just one example of, in this protein, we have in the DNA, we observe that the G at position 183 changes to an A, which then leads to that this valine at position 62 in the reference database changes to an isoleucine. So the triptych peptide is underlined where we have the valine. So the change we see is that we then instead get a modified peptide that then will have a different mass and a different fragmentation spectrum. But if we go through and take all the single nucleotide variants that we see in the genome and see which extra triptych peptides we get, we can then make a larger database. So this is probably the simplest change on the genomic level that then propagates to the proteome. We can have more complex changes. So for example, in this case, we have a more dramatic change where this, on the DNA, this C at position 155 changes to an A, and that means that the tyrosine changes to a stop codon. So what will happen then is that most of the protein will not able to be produced in our sample in the tumor. So we can only expect to see peptides from the first part. So of course this is much more difficult case in the sense that in proteomics, our coverage is limited, so if we don't see something, that's not proof that it's not there. So, but that's one example of things that can change. And so what people do then is to sort of evaluate all the possible effects that we can have. And Kelly is going to talk more about what kind of changes are, but I'll just quickly say that most of the different ones are related to splice variants that we can get from the RNA sequencing. So this one up here is just simply that we have 3 exons, and according to the reference database, exon 1 is connected to 2, which is connected to 3. But then in the RNA-seq, we also see that exon 1 is connected directly to exon 3. But we also see a lot of other cases where we get connections from the middle of an exon, from an intron, and so on. And so Kelly is going to talk more in detail about these things, what you can expect, and how to create these tumor-specific databases using combination of usually exome sequencing where we get variants and insertions, deletions, and the RNA-seq where we get the different splice variants. So what are the effects then that we see from these genomic changes? So it can be the protein sequence can change, as we looked at these 2 examples. We can also have that the modification site is changing. But we can also, in a lot of cases, have mutation that doesn't change the protein sequence, but changes its level. So it's either from a mutation you get more of the protein or less. The same for modifications, that mutations can lead to both increasing and decreasing of the mutations. So I have a question. So why do we care? What do we use these? So now we make a big catalog of modifications that we see on the genomic level and that we also see on the proteomic level. So anyone have any suggestions for what do we do after that, after we made this catalog? No? Yeah.
Like if you have that catalog, then we can, when we test our sample data set, and if you find their variants, and we can cross check with the catalog whether this variant is specific to cancer or not.
Yes, so we can look downstream and see if this, I mean, both that it's correlated with cancer or that maybe it happens in a tumor suppressor gene or an oncogene that changes things, yes. And another thing that we can, of course, look at is if these peptides are only present in cancer and not in normal samples. Then we can try to target them and see if that we can, for example, try to activate the immune system to attack cells that display these peptides, or in other ways do targeting. And there are examples of treatments like that already.
Or we can also use this ALE catalog to create a database which we can use for proteomics mass spec search.
Yes, no, so that's what we do, yeah. So, no, but I was thinking of after that, after we've done the, yeah.
My question is, as you were saying, so do you mean like peptide specific to cancer?
Yes, that's what I was thinking. So two ways. So either peptide specific to the tumor, or that a protein is activated that's usually not active, that is maybe not expressed at all in most cells, but gets, because of mutations, gets expressed. So that's another thing that we can do. So for example, in breast cancer, HER2 would be an example of that. In most cells, we have very low amounts, but in a subtype of breast tumors, we have it's very highly expressed on the surface of the cancer cells, so we can then have targeted treatment for that.
But we are doing the mass spec analysis, so we are getting a list of peptides. So like if we are saying this is cancer specific, but that protein is also there with the normal sample also. So I don't know, but how we can say any peptide protein, we can say, okay, this is cancer specific or not?
I mean, yeah, so that depends on your experimental design. So if you, for example, you'll have to then analyze a lot of tumors and also normal samples to see that in most of the normal samples you don't see it. And I mean, what one can do is, for example, look at studies like GTEx that looks at a lot of normal samples, because if for a good treatment you want that there is no expression in any of the organs, pretty much, and you can use these public data sets to compare. Another thing that we can do is to go back to the central dogma of the molecular biology, since we now are measuring these different kinds of molecules. So we are doing whole genome sequencing, we have RNA-seq data, we measure the protein levels, and we measure how much modifications we have. So that's in CPTAC, that's what we usually do. For some projects we have other types of measurements, but these at least basic measurements that we have. So we can look a little bit at how these relationships are. So for one, as was already mentioned several times, that the proteins and the modifications are the functional gene products. So that's what makes the phenotype. But then, of course, it's much easier to measure DNA and RNA. There are much more automated methods, and that still, even though there's been a lot of improvement, it's still rather much more difficult to measure proteins and modified proteins. So it's often, one can probably have, in most studies where one only looks at, for example, RNA-seq of tumors, these are many more samples are analyzed. So then the question is, okay, so it's cheaper and faster to measure RNA. Can we just measure RNA instead and not worry about the proteins and their modifications? So there are many arguments for why that's not a good idea. First of all, there's a lot of additional regulation than this. The other thing is that this process, the transcription and translation, is rather slow, but the modifications can be done, both added and removed, very fast. And then we have different degradation rates, usually for RNA and proteins. So we shouldn't be surprised that there are substantial differences between RNA and protein measurements. So if we just look at one example, so this is in breast cancer, it's ERBB2. So we look here, we have copy number and RNA levels. So we see that there is a group of samples that have a higher copy number of this gene, of ERBB2. And of course, this is the HER2 subtype. And we see that that's correlated with transcript levels. So we have more copies of the gene, we will have more RNA. So we have a nice correlation there. So if we go down and look at the translation, we see that also RNA is correlated with the protein here. So when we have many transcripts, we also have more protein. And finally, if we look at one of the phosphorylation sites, it's also very highly correlated with the protein levels. So that's what we, if there is no additional regulation, that's what we would expect. So we have more copies of the gene, we get more transcripts and more proteins. But if we look in the same gene in this example in another tumor type, so in ovarian cancer, first of all, we don't have really any copy number changes. All the samples have the same copy number. They pretty much have the same transcript levels also. But even though there is the same transcript level, we see quite a range of variation of the protein. Almost it's a little bit smaller than the range of proteins in breast cancer, but it's definitely very comparable. And finally, we don't see any correlation between protein and the same phosphopeptide, as we see so very strong correlation here. So these are observations that we see, and we can look at different genes, and we see large differences in between different tumor types, between different subtypes. Just to look at one more example, this is one of the keratins. So here we don't have any, so we're looking at breast cancer, ovarian cancer, and colon cancer, and we don't see any changes in copy number in any of the tumors. We see very large range, difference in range of the RNA levels. So in breast cancer, we have a very large range. In ovarian cancer, almost no change in transcript levels. And in colon cancer, somewhere in between. Then, if we look at comparing, looking at the translation, so between RNA and protein, breast cancer, again, we see correlation. And we see some correlation, but the range of RNA is very small for ovarian cancer. And even though we have quite a bit of variation of the transcript levels, there is no, the protein levels are constant. So we see that in this case we have very different regulation of both the transcript and the protein. And so if we look, so these are just two examples. So now if we look more globally, we see that there is a wide range of, so this is copy number transcript. We see that there are some that are highly correlated, others are even anti-correlated, and we see this wide range. And for every comparison, both transcript protein and protein phosphoprotein. So what does it mean now? So we see, if we see a correlation in one case, we don't see a correlation. How do we start thinking about this? Any suggestions? Maybe we can start, let's say, what do we say if we see a anyone?
So the reason can be the DNA is not transcribed into RNA, or some part of the DNA is not transcribed into RNA, and the RNA is not, might be the RNA is translated into protein. Some of the protein has been degraded and some of them is used in the downstream processes.
Yes. So, and we can also have a case where the proteins are produced somewhere else in the body, like we'll also see a lot of blood proteins that will be in the tumor, but there is no RNA there because it's being produced somewhere else. So there are of all these things, and we have the degradation, so we have a lot of different things going on.
Ribosic acid proxy for translation.
Yes, we can, yeah, so riboseq would be somewhere in between here. It would measure the actual translation. It wouldn't measure the amount of protein that's there, but it would measure how much is actually translated at the moment. So that, yeah, that's definitely, that would be going a step in between the RNA and the protein.
In today's lecture, you got a very broad, detailed glimpse of how proteogenomics could help to reduce changes in protein sequences due to the mutations in the gene, how to identify the changes in modification sites of proteins, also in which way proteogenomics could help to understand the changes in the level of protein expression due to change in the gene sequences. In diseases which are characterized by the changes in the protein sequence due to mutations at the genetic level, the proteogenomic analysis could help to provide us the development of disease specific databases where the modified protein sequence information could be made available. Taking the example of ERBB2 and using proteogenomics, Dr. Fenio showed that there exists a clear correlation between RNA, protein and phosphoprotein expression in breast cancer. However, same did not hold true for ovarian cancer cases, as there was only correlation between RNA and protein, but not the phosphoprotein levels. So, you can see that there is no clear pattern depending on each disease, specific context, the correlations could actually vary, and therefore, these analyses on individual data set by looking at specific questions are very relevant. Though proteogenomic studies may not always show a direct correlation at all the levels, they still offer and provide information which could be used to answer questions which are very relevant to disease pathobiology. In the next lecture, you will be introduced to few more concepts of proteogenomics in clinical studies by Dr. David Fenio. Thank you.